Hello again. Today I will be talking about how the DNA replication in prokaryotes occur. I'm using a linear to represent how DNA replicates. We will zoom in to the replication fork of the DNA structure. First, let's go to the origin of replication, or where the DNA replication begins. The next step is represented by the blue hook. Helicase is the enzyme that unzips and separates the DNA. Note that the sugar molecules, or phosphodiester bonds, are the backbone of DNA, and they are not broken. It is just the hydrogen bonds that break. Next, the light red dots represent single-stranded binding protein, which binds to the strands to keep both separated and to prevent any coiling. Also, the clear band on the left side of the structure represents gyrase, which removes the strain in front of the fork while keeping the unzipping from happening too fast. RNA primase makes primers on both unwound DNA strands, as shown by the yellow and purple tabs. DNA polymerase III then comes in and builds the new strands in toward the 5' direction. DNA polymerase III bonds to, in our case, the A, which is in the 5' direction. On the top strand, it is a continuous connection, which means it is the leading strand. On the bottom strand, notice the D square had to make multiple stops before reaching the A square. These stops represent the repeated synthesis, also called Okazaki fragments. The strand that has repeated Okazaki fragments is the lagging strand. DNA polymerase I comes in and replaces the RNA primer, or the slide from D to A, with nucleotides on the leading strand. Without this, the DNA processes would not be coded correctly and could end up missing a very important thing, like an important protein void from the DNA structure because it would still be RNA. Lastly, the lagging strand. The Okazaki fragments are sealed with the enzyme ligase, which you can see represented by the green line connecting the stops.